Welcome, 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 everyone. We're going to go ahead and get started. Thank you so much for being here tonight for our Science on Tap. This is a very exciting evening, and we have a great speaker for you. But I wanted to just do a couple of logistics and make some announcements. First, my name is Cinnamon Moffat, and I will be your host for this evening. This is a hybrid event, so that means we have folks in the room and we have folks online. If you haven't been to one of these events, there's a couple of things that make it a little easier for everybody to engage. For folks online, we have our volunteer, Roseanne. Hi, Roseanne. So if you put any questions you might have, technical issues or questions for today's speaker into the chat box, Roseanne will make sure to address those or read those out to our speaker at the end of our presentations. But thank you all for being here. Other logistics for our hybrid folks: because this is a webinar, your cameras, mics, and screen shares are disabled. So the way you can talk to us is through that chat box, but please engage. That would be wonderful. For folks that are here in the room, because we have folks online that cannot hear unless we have a mic, when you ask your questions, if you just raise your hand, I will run around and make sure you have a mic so that folks can hear. It also helps all of us hear your questions as well. So that's kind of the way it works. But again, thank you so much for being here at our Science on Tap. If you are new to these events and you would like to get information about when we have them, there is a sign-up sheet behind Bob, who is our speaker tonight. You can put that on your email. The only reason we use it are for these public events, so you're welcome to do that. Otherwise, you can go to the HMSC website, go to the events page, find all the information for our events there. A couple of things that I just want to make announcements about, and I'm gonna leave the box, Bob. I'm gonna go off camera, everybody. I'm gonna pick up something, and I'll come back, I promise. I wanted to promote Marine Science Day, which is coming up. Very exciting day for us on April 12th. This is Hatfield's open house. We throw open our doors and everybody puts out all of the great stuff that they're working on. It is a free event for all family, all learners. And so we encourage you to come on April 12th. That was a great day. The other thing I wanted to promo is that next month we have also a very exciting Science on Tap. Tuba Oscar Haller is going to be talking about changing science around sneaker waves. And after yesterday, I think we can all learn a little bit more about sneaker waves. So I'm hoping that you come back. That particular event, I got to double check, is on March 12th. So hopefully you all can come back on that one. That is in this room at 6 o'clock again. So Science on Tap and Marine Science Day. But the reason you're all here is for today's speaker. So I'd like to introduce Dr. Bob Cowen. Bob is the Director and AVP for Marine Research and Operations here at Oregon State University and Hatfield. Bob is a biological oceanographer who research centers on ecological and life history of fishes, particularly early life histories of fishes. And his work is extensively on the biological and physical oceanography processes affecting retention, transportation of larval fishes at sea in terms of examining larval dynamics, population replenishment, and conductivity. Bob is a co-PI of the Plankton Ecology Lab, where our research includes specific aspects of ecology of larval fishes and includes vertical migration, growth, survival, and feeding. And I think we're going to learn about all sorts of that stuff today. So we're so excited that Bob is here. Bob, can you make sure your mic is on? And I'll hand it off to him.
Green. Okay, we're on. All right. Celebrate science, right? First, those who are in the room, you look around the room, you don't see Sue. Unfortunately, Sue has been walking around since Thanksgiving on a broken ankle, and it was just finally diagnosed. So she's not walking around anymore, and she couldn't come here tonight. And I do think still the nice thing of having her name here is it actually had a larger draw than if it was just me. So, all right. So let's also, one thing I want to note is the work that we're talking about is the work that comes out of the Plankton Ecology Lab. And there are a whole bunch of additional people. I started listing them here and realized I didn't have any room. Throughout the talk, I'll be referring to some of their work. And at the end, I will also show more names, but there's been a lot of great collaborations that we've had a chance to work with, and many wonderful students and postdocs. And I thank them all for getting us to this point. So I start off with this. It is a remarkable animal. This is a female blue marlin in the Atlantic with a very unfortunate end to its spectacular life. But I bring this up to remind us that all great beasts, and here I'm referring to fish, that's just my bias, are starting off really small. This is a baby blue marlin. This turned into that. And it's important to sort of think about this because it's the survival of these stages, these little stages, that actually have a mark on the population, that affect the year class, the size of the year class. And so the processes which are driving this survival are they're largely spatially distributed, meaning there's variation with space. And that variation plays a huge role in sort of the spatial outcomes of where we actually see larger year class or smaller year class. And we can look at that temporally, but also spatially. And the difference in size also points to the fact that the processes, the things that they're being affected by also vary by scale. And so we need to look at these things across sometimes really large spatial scales. Sue and I have been working for a long time on the processes of larval transport, and not just how they physically get to somewhere, but how they manage to do it and survive. So that affects environment, that affects growth, it affects their prey interactions and predator interactions. And here, this is a typical life cycle of what we call bipartite life cycle of a lot of coastal fish, a lot of coastal inverts as well, where the adults tend to be relatively sedentary in a nearshore environment, whether it's a reef or a sediment area. But the young, the larvae, should be able to do this. Hang on. No, I guess I'm not able to do it. And nope, I do. I need a laser pointer. There we go. So the larvae are offshore. So when they spawn, the eggs and the larvae get carried offshore. And so entirely different environment than where the adults live. And at the end of that larval stage, the successful ones are transported, swim. We don't know completely how they get into shore. And then they start the rest of their life as juveniles into adults until they reproduce again. So what our work does in the lab, the areas that we are looking at are, as they're going through these larval stages, what are the environmental conditions that they're encountering? How are they responding to that? Are they driving the distribution? What about their prey fields? What are they encountering? Is a good year is likely going to have a lot of food. Bad year is likely not to have a lot of food, or it might have a lot of predators. So how do we look at all of those things simultaneously? Well, we have a couple tools that are available to us. Three primary ones. First of all, on our cruises, we can collect with nets. We have a couple different net systems. One that we're particularly fond of is the mock nest. And we have what we call an asymmetrical one that has small nets that are for inverts and plankton. And the other one is for the fish. It's a much larger one. And this gives us an ability to sample different vertical discrete layers of the ocean. So it gives us a little two-dimensional, horizontal on the surface and vertically. It also provides us, that's not working, samples in hand where we can pull otoliths from, and most of you have heard about otoliths, but it's a means of measuring their growth rates and their age. And we can relate that to things such as their diet, what they're eating, how that might be varying through time. And then finally, our third tool is to kind of look at the broader background environment. So what does the physical environment look like? And then what does the whole planktonic environment look like in terms of organisms, prey, predators, the fish themselves, and how are they interacting? And it's this last point here, this approach, which we use an in situ measurement tool that I'm going to be focusing the rest of this talk on. So I'd like to step back and talk a little bit about the evolution of sampling in the ocean. Very early on, it was pretty simple. A ship's captain would estimate the drift of their ship in different locations, so how fast it was moving and in what direction. And then over time, they have enough samples, they could start seeing that this is an area where maybe we're moving faster. And then coupling that with temperature, surface temperature, very simply collected by a bucket of water and a thermometer, could start putting some of this together. And one of the first maps was by Ben Franklin and Timothy Folger of the Gulf Stream. And, you know, it's crude, but it's actually rather remarkable, right? And this is in the early or mid 1700s. Fast forward about 2 centuries, and we had a lot more technology to work with. I'm going to ask show of age among our scientists here, how many have actually worked with one of these? Yeah, there we go. So this is a water sampler and reversing thermometer, and it was pretty cool when I was a graduate student because it enabled us to get an idea not just of what the temperature and with the water sample, the salinity, but by depth. And for those who haven't used this or don't know about it, is you can see this guy, he's attaching it to a cable that's going down and sends that down, and every 10 meters or 50 meters, he's putting a new one on as that thing's getting deeper and deeper, the first one. And once they have them all on, sends a little messenger down, a little weight that wraps around it. And when that weight hits here, this thing, the top detaches from the cable, swings around, and then it hangs upside down. And that does 2 things. One, it freezes the mercury at one spot, so when it's upside down, it's a reversing thermometer. You can tell what the temperature was at that depth of water. And the second thing, once it's upside down, is it releases another weight, and that goes down to the next one and starts it over and over and over. Once that's all done, then you got to pull it up and you take each one off one at a time. And you know, it was pretty time consuming to get a tow. A whole cruise would end up with something like this, a transect every 10 or 20 miles offshore, and you can see samples down to about 500 meters, but it gives a nice view of how temperature changes with depth. So a lot of work to get just the temperature and salinity, right? Well, fast forward another couple of years, and technology advances significantly. Now we have tools like this whole combination of instruments here that, first of all, the CTD gives us a digital reading, connectivity, and temperature with depth that enables us to, and it's sampling at about 40 hertz or 40 times a second. So it's just getting really fine data. Other sensors on there can be oxygen, temperature, fluorometry, whatever. And so you're getting all that information just by dropping this down and pulling it up, and it's coming off digitally such that on the ship, look what you can do. You can graph this stuff up. It's beautiful, right? It's amazing. Somewhere along the lines, an ADCP was developed, which gave you also the ability to look under the water and see all the different directions the water was flowing, the currents. So this is a point. As a larval fish biologist, I started to get a little envious of these physical oceanographers. I had the opportunity as a fish biologist to collect all these nets, samples, and maybe on a cruise we'll come home with about 200, 250 samples. Every one of those had to be strained, sorted, larval fish picked out, ID'd, and finally at some point you have data. We put that all together, and then we go walking back with our data to our physical oceanography colleagues. And this is like 4 years later. And they're going, oh, wait, I've already published on this. I'm in 3 projects from them. I don't even remember what we were doing. So I'm kind of spiraling. And not only is the on-site sampling getting better, we have got models that are modeling the ocean, satellite imageries that are verifying this. And we're seeing a tremendous amount of variability out there. And how do you sample that across all these scales, from really small scales to something like one of these warm core eddies, cold core eddies or warm core eddies here? Just to sample that sufficiently with nets would take a really long time, a big investment. So here I go. And it wasn't until I was at a meeting in Stony Brook, New York. It was a larval biology, the first larval biology meeting that was hosted by Steve Morgan, Craig Young, and myself. And Cabell Davis gave a talk, early talk on the design of the VPR, the video plankton recorder. And this was an instrument that they were towing through the water, and it was photographing plankton, and particularly diatoms and copepods, those really fine resolution. And I was just going, all right, I found it. And so I go running up to him after the talk and say, I know this is a larval biology invert, but what about the fish? I mean, let's talk about something that matters. And he said, okay, I've looked at about 150,000 images myself personally, and I have seen exactly 0 fish. Whew, here I go again. But I'm thinking, wait a minute, no, there's a reason for this. And I talked to him a little bit more about the design, and it turned out that they were photographing a parcel of water about that big. And what I realized is fish are really rare in the ocean. So this is many of you have seen different versions of this, but this is a sort of abundance versus size of plankton. And you can see when things are really small, down in the micrometer or less, like viruses, they're hugely abundant. This is the number per cubic meter. So 10 to 13, I don't know what that number is. Is it a gazillion? It's huge. They're so dense. That would mean the amount of water you would need to sample them and get a large sample was just minuscule. But when you get down to things like dinoflagellate, diatoms, copepods, you're looking at a little more reasonable, 1,000 to maybe 1,000,000 per cubic meter. Again, that allows you to take a sample like that. And that's why the VPR was working so well. But if you're interested in something as cool as larval fish, here they are down here. On average, there's about 1 larval fish per cubic meter. And so 150,000 of these is barely a cubic meter. And so that's why they weren't seeing any fish. And even if they saw one, as we all know, a sample size of 1 is not going to get you very far as a publication, let alone understanding things. So we had to find a way to sample down at this low density. That's why our nets are so big. So you have to filter a large volume of water. So we looked at different ways that things are being done in this small size class. There were in situ molecular approaches for identifying just the genetics of what's out there, and flow cytometry has been developed, which is really cool. But again, really small volumes of water are being sampled. The VPR and other instruments now are out there that are sampling in that size range that the original one was. That's capturing well the diatoms, dinoflagellates, the primary producers, and the copepods. So it's getting the sort of the base of the food chain, which and describing quite well. But I was interested in this stuff, the larval fish, some of the potential predators and some of their other prey, including copepods. And how do you get, we're still looking at 4 orders of magnitude difference in size. How do you get a large enough volume to sample that? And still, sample it such that you can see the larval fish themselves and potentially some of these others. So we had to look at a approach to doing this. And I had a fortunate having a technician who was very much into photography and a friend of his who was a marine engineer. And Cedric and Charles and I were able to sit down and start talking about design. And we found a approach which is called shadowgraphy. And that's where basically you shine a light and anything that passes through that light creates a shadow. So here you can see my shadow from this light, right? It's a little blurry. But it is what's really interesting thing about that is if your light beam is small enough, here we start with an LED and then we focus it through a little pinhole, which is about 5 microns. That significantly, the physics significantly increases the depth of field. So if I'm over here making a nice shadow puppet, let's say against the back wall, I'm not able to do that here, or I'm all the way over here, it's all in focus. Whereas the VPR and any of you with a camera, if you're trying to do macro photography, it's reflective light and you're trying to find that depth of field and it's really small. And that's what the VPR was using, which works perfectly for their purpose. But we had to go with this shadowgraphy. So let's follow the light beam. LED, single LED, low level of light through a condenser that focuses on this pinhole. And then as that light comes out, it's spreading out. So we run it through this lens called a plano convex lens, and that straightens it out so they're now parallel light beams. And I'll explain that in a moment. But those hit the mirror, and then they bounce out through a window, go across about 45 centimeters of water, back in, hits a mirror, and you notice that they're all parallel. That gets rid of parallax, because if an organism was at the first window coming out, it'd be this big, but with parallax, it would look this big if it was at this end. And so we had to make them really parallel. And so that enables us to get size information. And then we would refocus it back down to this camera. And this is, excuse me, this is a line scan camera, which is kind of like a Xerox machine. It's scanning line at a time as, in the case of a Xerox machine, the paper is moving over it, and it builds a full picture. Only this camera is scanning 36,000 times per second. And as we tow it at 5 knots, every scan at that speed corresponds with 50 microns. And so we start putting together a picture that is resolution down to 50 microns, but it's being built out. And if we tow it across this room, we have one single image across this room. If we tow it for 10 miles, we have a 10-mile long image. And you can see everything in respect to each other in that. So this is sort of how it works. So the light sheet here, the yellow, is the vehicle, and it's moving this way. And the fish here is going between that beam of light from one window to the next. And as it passes, it's picking up the shadow, and it basically builds that picture. So and the really cool thing that you're going to see in some of these images is it turns out most of this plankton is translucent. And so you actually start seeing through a lot of these things. You can pick up different aspects of them, and some of them are remarkable. It really changed my enthusiasm on one hand for some of this. So again, scanning 2,000 times, or each scan is 2,000 pixels. There are some cameras which go way up, which we just couldn't handle that much data. 36,000 times a second. So I already gave you all that. Never mind. So this is the second version of the instrument. The camera light beam is here. It comes out across, comes out across, this is that 45 centimeters opening. And as this thing is towed through the water, anything that passes between those two pods is what's being imaged. Then these upper parts, there's a float to keep it so it's stable. We have one pod here that's got all the brains of the whole system, and the other one was full of other sensors. So we had temperature, salinity, oxygen, pH, fluorometry. And so every image we saw, we have a full description of the physical environment that it was encountering. So we're combining that all, as, as we're doing these tows anywhere we do them. The newer version, which actually we've developed while we're out here, working out here, had two primary criteria. So it's basically the camera is somewhat the same, but the cage that it's in now is different. And that's partly because two things. We encounter lots of crab traps out here, and that other vehicle was just catching them and just holding on to them and getting its arms ripped off. It was a mess. This one actually sheds them the way design is. There's no hard edges. The cable just comes around our tow cable, and it just fully pulls itself off. And the other side is, it looks like a Jeep or a dune buggy, and it's because we actually want to work very close to the bottom at times, and for the occasional, we want to make sure it doesn't disappear and break up. So this is capable of that, and I'll show you how close we get sometimes. And this is launching from one of our vessels. This is the Langseth that was out there. So what's this camera see? This is where I was just amazed. Here we have, these are our ctenophores, comb jellies, and you can see, I like this picture right here, the translucent nature of it. So you can see a lot of its primary organs, and you can see musculature and nerve networks on this thing. And this is in situ. This is just in its natural environment. So how their tentacles are laid out, or how they're swimming, the undulations that they were doing. Larger things come through as well, such as scyphozoans, the typical jellyfish, and seeing how, again, they lay out their tentacles. Because we've modeled this, and we've done it in a flume. There's no turbulence in the area of the camera where it's seen. The turbulence sort of develops a little bit further back, the breaking point. And so we're seeing things unaffected by the presence of this vehicle. These are small hydrozoans and the pelagic stages of them, really delicate. See the gonads on this guy, just spectacular images in my mind. These are siphonophores. These are colonial animals, for those who don't know, and they have feeding. There are colonies. They have feeding groups. They've got the tentacle cells. They've got reproductive components, and some of them are kind of chaotic in the way they lay out their tentacles. Some are really organized, and we see these repeatedly. So this is not just a once in a life, one thing. It's really remarkable how they lay out. Crustaceans. Look at this copepod. We're seeing not just the antennae, but the secondary antennae. You can see the eggs there. This is a larval lobster. So these are tropical. I can really tell they're tropical because this guy in his beach chair. That's a stomatopod. This is a particularly cool one. Some of you may know this animal, Phronema. It's an amphipod that is parasitic. It gets into a salp, which you can see it in the salp right there, and it will lay its eggs in there, and then the young will feed on the salp. But this was an inspirational animal. You can see it's beautiful. Its mouth, its eyes, its secondary. These are our feeding appendages. This was an inspiration, the inspiration for Alien. And in fact, the producer, whoever wrote it, actually had a marine biology friend that showed that to him. So this was truly the inspiration. Apiticularians, they're super abundant. They're super important for diets of a lot of fish, particularly in more tropical areas, the tunas. Miriam knows that well, are an animal that creates sometimes they're free in the water, but often they have mucus homes that they build. They build about 7 of these a day that are used to trap particulates in the water. They can filter in, and they can also consume once their house gets really, really dirty. And unlike us, we either continue to live in the dirt or we occasionally clean it. They just trash the house and go build a new one. So they do that, as I said, about 7 times a day. But these just spectacular. Try and bring that up in a net, and it's just mush. So, and then finally the fish. We do see fish with this. We see a lot of fish, and they're really spectacular. Again, seen through some of them, when they're that translucent, like this flatfish. You can count all the fin rays. You know, very definitive. You can see the gut. I'm hoping at some point to actually see the otoliths at someday. Anyone know well, if you are an ichthyologist, but this is a larval swordfish. Being able to see some of the fine details of the little denticles along the base of it. Well fitted. You can see this swim bladder in this cyclothony. You can see how they're swimming. How the trailing, you know, we get these in a net, and first of all, they're usually broken off, these filaments, but if you do have them, we're not quite sure how they use them in the wild. So we can see that. We can see this fish, Leptocephalus, swims with its whole body in an undulation, whereas this one is just its fin that's and it holds its body perfectly straight. Sometimes we're so fortunate, like on this myctophid, where we can actually see the pigment, and this enables us to identify it, in this case, a species. Just some really spectacular things. This one, the Leiopropoma, long filaments, and they look like the siphonophore tentacles. So they're mimicking in that. We've seen lots of the lobster larvae sitting on top of some of the ctenophores, just hitching a ride. They're not eating them. They're so all sorts of stuff that you wouldn't normally see. And this is one of the last fish I want to show you. It's a Leptocephalus larvae, so it's an eel larvae, and it doesn't come with all these numbers on it. We put those there, just in case you're wondering. But you can see the incredible resolution to where we can count every vertebra or myomere. And so here they are, they're numbered, and these are things that you use to identify these fish. You also look at how many gut loops they have. Well, there are 2. And then you see how long is the gut. So where's the anus? It's right there. And that tells you how many preanal myomeres there are. And that helps us set it down between these 2 species. And we're also able to see a connection between the kidney and the anus. I mean, it's just to see this stuff under a microscope is amazing, but to see this is a fish that was in situ, just zipping by, and it's still out there somewhere, probably as a big eel now. So pretty cool. Well, it inspired me just as a natural history perspective to see all this stuff, and it sort of re-energized me a little bit. But obviously, this was built for a purpose, and there are lots of different applications. And one from a teaching perspective is getting a little bit of a gestalt of what these different pelagic environments look like. If you're a diver, you know what a clear water day looks like and a murky day. Sometimes you see all sorts of stuff in the water that I call whale snot, and sometimes you just have these beautiful vistas. Well, you can get the same thing from this. This is an image from Stellwagen Bank, which is up in the Pacific Northeast, Atlantic Northwest, so off of Connecticut and up near Massachusetts. And this is a fairly productive environment. And you can see in here these stringy, really thin strings, like right there. Those are diatom chains, and the rest of these little spots are copepods. So this is a really productive area, and it's dominated by this pretty short food chain, diatom to copepod. Compare that to a place like inshore Monterey, this is a shallow area, very calm, very productive. In fact, it's so productive, and I'll show you some other data from here, where these thin layers set up because of these huge algal blooms of Pseudonychia, which is a toxic um, plant plankton. Sorry, but and as it's near the end of its planktonic bloom, it starts exuding all this polysaccharides and creates these flocks, like these little clumps, and they start coming together. And so this is a lot of productivity in this big bloom, and you're just catching it at one point. Here's the Gulf of Mexico, generally fairly oligotrophic, but this is getting a little closer to the Mississippi River, so there's some more nutrients there. And you can, so if you think of the Stellwagen Bank, you can see the copepods, and if you really squint, you can see the diatoms. Just have to trust me on that. But it's not as dense as Stellwagen. And then you look at a place like the Florida Straits, which is very oligotrophic, and you don't see a lot. It's very clear water, very little in the background, very little noise, in fact, in terms of that. They're still countable. We can see them in the imagery when we focus on it, but it gives you this sense of how different these places are. Another thing is the scale of which patches occur. We know fish school, but what did plankton do? Here's a single image, and I should explain a single image. As that's scanning, each scan is 14 centimeters long. That's just the way it worked out. And it's building an image, continuous image, but as soon as it travels 14 centimeters horizontally, we cut it as an image. So the next one is just, you can tie these all together, and it's just a continuous. So just for the storage purposes, we do a frame at a time. So that's what you're seeing here. And here in one frame, we have 9 crabzoea, all oriented the same way. So, that's a dense association in my mind. This kind of went on for a while in these images, but I'm just making a point with a single one, how close they are. And then here we've got 2 images together, and these are a type of larval fish, chlorophthalmus, and they're all oriented. And this is 2 images, I said 25 centimeters of travel, and that comes out to a local density of about 308 fish per cubic meter. Remember I said sort of the average is about 1 per cubic meter? So this is, if you were to take that as a larger 1 cubic meter volume, you would have a huge number of fish. So this is a really dense, for fish, this is super dense. Put that in perspective, this is a little jellyfish, Narcomedusa, and same distance, 2 of them together, a lot more a lot more animals. And this is about an order of magnitude more. This is about 5,200 per cubic meter if you sort of spread that out. But sometimes things get really dense. And sometimes things don't work. Oh, turn off my pen, thank you. There we go. Okay. So this is moving along, and this is just 9 of those images. So this is about 1 meter long. And I have not counted all these, but my guess is we're looking at over 50,000 per cubic meter here. You can see it just gets denser and denser and denser. Also, if you look at it, they're mostly oriented the same way, which is kind of interesting. So we can look at patches another way. Here we've taken across a whole transect of imagery, and we looked every time we found a fish. But you can do this for any taxon. We looked how far was it to the next one, and you can start looking at doing what's called a nearest neighbor analysis. And this red line is what it would look like if it was just purely random distribution. If you're above that, then you're actually having clumping. And so we actually found that typical patch size for fish is about 3 to 10 meters. That's really kind of remarkable, actually. It's to me, that's sort of suggesting that larvae, even at that scale, say here to the wall, they're aggregating, or they're being aggregated. Don't know which, but it's kind of interesting. And then we can take this to a much different perspective, a slightly larger scale, and look at all of our data at once. And here what we did is, this is off the coast of Florida, where we had transects, Miriam was on this cruise. We took 10 kilometers of single horizontal depth at 10, 30, and 50 meters on the shelf, or it's a very narrow shelf, but near shore, and then out in the middle of the Gulf Stream. And so we have continental and oceanic here. Shallow is 10 meters, mid depth is 30 meters. I'm not showing the 50-meter one, and then these are all the different taxa that are identified in this instrument. And color is density. It's a log density. So orange are really abundant, like diatoms, protists, Trichodesmia. And then you look at some of these. So they're so ubiquitous that it's just one big patch. It's all over the place. It's completely singular. But there are other things like these tunicates or salps or the fish, some of the decapods, that are really quite patchy. And you find these really distinct patches, then nothing for a while, another distinct patch. Trying to sort out what might be driving these things. We're still pretty early in that, but it's the ability to see so many different scales of patchiness for so many taxa in the same data set, it was kind of interesting. And then comparing between the two. And it turned out that the size of the organism was driving the degree of patchiness. The smaller organisms were more ubiquitous, and they tended to be more independent, apparently, of the environment. And they were just present everywhere. Versus smaller things tended to be more patchy. So something, what's driving that is suggesting that there might be more of a biological component. And then we can look at it as a different way and actually compare specific distributions to each other. And this is in the same general area in the Straits of Florida. They have these, as the Gulf Stream is cruising along, it's dragging some of that continental water, and it creates these little sort of submesoscale eddies, and they spin for about 5, 6 days, 7 days. And so we were sampling across one of these eddies, and there's eddy water, there was sort of the frontal interface, and then the Florida current. And we've looked at this for a lot of different taxa, but there were two that were sort of stood out by their similarity. This was the fish larvae, and down here was a ointhona, which is a small copepod, which actually was very prevalent in the diet of a lot of these fish. And so you look at that and you think, oh, it must be the fish are responding to the presence of the copepods, because they're really quite similar. You look at these hot spots here, hot spots there and there. But Moritz Mo Schmid did a nice study on this, random forest models. And basically, we found that 80% of the distribution could be explained based on a couple predominantly physical variables. The presence, if you took Ointhona out of the analysis for the presence of larval fish, it only dropped the percent explained by a little around 2%. So overall, it was 80% was the physical variables, and the Ointhona was primarily driven by physical environment. So for whatever reason, these two taxa were being aggregated by a physical system, not directly related to each other. You can look at this again for a variety of different groups. We can look at fine scale vertical distributions, which is really quite interesting. Remember I mentioned the net systems. we're able to sample 10, 20, 25 meter depth bins. What we're looking at here are 1 meter depth bins, and we've aggregated our data to get to that. We could actually look at 10 centimeter or 1 centimeter depth distributions if we wanted to, but we've aggregated this into 1 meter, and we're looking at the presence, the depth-related presence of larval fish, and we find 3, 4, and 5 meters deep is a peak, and that's exactly where the peak of the copepods were. Overall pattern was somewhat similar, but the fish are clearly closer to the surface, but they really are concentrating where the fish are, where the prey are concentrated. This also was at Stellwagen. Now I showed you the Monterey Bay. That was an area where these thin layers set up because it's super cool, calm water. It's only about 16 meters deep. And it's predictable enough that we're actually able to get NSF funding to go and study thin layers at this place. That tells you how predictable it is. And so what we're showing you here is the distribution of individual flocks of Pseudonychia. And I'll show you pictures of these. And what we've done is measured each one based on the long axis and the short axis. So it's long over short. And if it's something that's really elongate, it's going to have a high number because it's a long axis over a short one. If it's globular, it's going to have a 1. So that's sort of the range we're looking at. And if you look at this in the shallow waters down to about the pycnocline, by the way, is about 6 meters deep. That's the density layer there. And in the shallow water, we had a fair number of these fairly elongate flocks. And this is what it looked like. So these are these individual flocks, and they're sinking. They're all oriented in the same way, and they're all going down. When you get to the density layer, everything is stopping because they're in less dense water up above. They're mostly water themselves. And as they get down there, it's going to take a while for that density to calibrate. And so they're aggregating at that density layer, and they're slowing up, so they're becoming more round. And so you see they have very low ratio. And they're concentrating there, so they're actually, that's where our highest chlorophyll is. And then down below, you have a lot lower chlorophyll, and just because a few of them are popping through from the mid layer. So this is the 3 different layers, and you can see more active, stretched out sinking, accumulation and retention, and then occasionally something popping through. Very interesting to see that mechanism. Oh, did that again. This is just a time series across out here. Actually, this is in Southern California, I believe, because it's Jessica's work. And you can see as we're going from, say, 3 in the afternoon, if I get this right, 3 in the morning, excuse me. No, 3 in the afternoon. Here's the afternoon over here. So 3 in the morning, euphausids are high up in the water column. You can see just this high number here. And then as the sun's starting to come up, you can see the numbers starting to drop. And during daylight hours, there are relatively few. And then after sunset, around 1900, they're starting to come back up again. So there's a really clear diurnal vertical migration, and actually able to sort of see that pretty distinctly. We can also look at the animals themselves and get a sense of orientation again. And then this is one last thing, which I was sort of intrigued with. This is back in Monterey. We had been sampling this. Basically, the study was to look at the formation and sort of the process of building a thin layer and then seeing what happens as it starts to degrade. So we were sampling this area, relatively small, very dense sampling, for about a 8-day period. So from June 30th to July 7th. And each day that we were sampling, we saw a lot of these Bolenopsis, which is a predatory ctenophore, fairly aggressive predator in the plankton world. And we measured every single one we saw. And so this is a sort of a population measurement of what the size of the Bolenopsis was. And we actually saw over the 7-8 day period about a 4 millimeter increase in that population. So it was a longitudinal sampling, not the same individuals, but just the population. And there was very little movement around there. And those growth rates actually correspond to those measured in laboratories. So it was, you know, seems like this was a pretty good measure of that. And it's kind of remarkable, again, from in situ data to have something of that resolution, be able to do that. Finally, we also had orientation. That Bolenopsis, 0, by the way, even though it's pointing down, is upward in this. So the Bolenopsis, almost all of them were always swimming in an upward fashion, almost 100%. Chlorobrachia, the one on the right, was generally trying to go down away from Bolenopsis, apparently. And you can sort of, they weren't quite as good at directing it, but it's predominantly in a downward fashion. So orientation, spatial distribution, interrelationship between all these species, we're getting a lot of capacity out of this instrument. So remember, if I started off that the time it was taking, you know, to analyze one cruise worth of samples sometimes could take multiple years. And when we actually worked it out with people, we were talking 20 man years, man and woman years. Huge amount of time. And so my goal was to be like a physical oceanographer and walk off a boat with data. One of these transects that I showed you produces somewhere on the order of 250 to 500 million images. And we're going, how are we going to identify that? So I went out of the kettle into the boiling pot, whatever the saying is. We were suddenly inundated with data, imagery data that we had to analyze before we could even get to the data that we wanted from this. And so we've been working on a imaging processing. This has been a long haul with this, but we've been working with some of our folks like Moritz, actually a whole group of people here, but he published on this pipeline where you take the raw image and after a little correction on it, you segment it. And by segmenting, we take each of these individual regions of interest, even these really tiny ones, what we're just showing as an example, and we put them into little vignettes. And so this is where we would have hundreds of millions of these. On a whole cruise, we would have sometimes over 3 or 4 billion. So this is a huge data processing issue. And then we developed a AI, we've been doing this for a bit, convolutional neural network that would enable us to identify these different segments into as many as 170 different classes. That's not taxa, because we might take a fish, this is one orientation, so that would be one class, but fish look very different to the computer when it's pointed this way, so that'd be another class, and that might be a third, and then we would know to aggregate those as to fish or whatever it might be. So doing that, we end up with about 70 different taxa, and this whole process has gone from being a fairly slow approach to it, depending on the computing power we can throw at it, where now we're working with supercomputing centers in Philadelphia, and this stuff is going through at a much more rapid rate. But our goal was to get to near real time, where we're actually analyzing this stuff out at sea. And so we did a proof of concept study out here. We're taking our, this is what the data looks like when it's coming up. What happened to that one? Oh, it's not showing. Okay. Where all of our sensors, every time the instrument goes down or up, it plots the data. And so we have a plot for oxygen, temperature, salinity, fluorometry, all those things. But here, just taking a focus at oxygen, because we're interested in hypoxia, low oxygen, and we see it gets down into hypoxic areas down really close to the bottom, or down to 100 meters as deep as we go. But as we're coming along, you can see the bottom coming up here. And remember the design, we're getting really close to the bottom here. This is like a meter or half meter off the bottom sometimes. And it's fairly rugose. So we're looking at meter or 2 meter bottom like this. So it's like, where are we here? But we're getting very close. But we wanted to take this as an example. What if we were really focused on this sort of region? Where was the oxygen the lowest? What organisms were there? Where should we go and more explicitly sample? So we actually, with Chris Sullivan at CQLS, got us access to this very fast server, edge server, brought it out to sea with us, and we had Dominic was a undergraduate, a superstar computing guy, and he pulled together a program that would aggregate. This is our real-time data coming up here, and you can see some of the other environmental data, but it would aggregate it and actually run it through that pipeline. It was subsampling the data, but in near real time, we were sampling right off of Oregon here at the point that where data is showing here. This is 64 million images analyzed, not 600 million. So it was just like 10% of what we had. And it was laying out a indication of the abundance of different taxa. And in this case, we're looking at, for phytoplankton, it's also plotting the average distribution of them by depth. So this was happening while we were out at sea sampling. It was probably 10 or 12 hours behind the time of when we'd actually done that particular transect, but that's still time that enables you to respond to something that might be going on. So what are we doing else now? We've upped our research vessel significantly. And we have a project working now with some colleagues in ag sciences, Tiffany Garcia and Betsy Bancroft from Gazanga, looking at freshwater plankton in these ephemeral or vernal ponds and seeing how the populations are changing through time. And so we've developed this little mini version of ISIIS, hand carryable. It has a little entryway here, but we're not able to tow it fast enough. So instead we have a pump that pulls the plankton through it. So it's not quite in situ totally because things could be spinning, but here's some imagery on the right here that you can see of copepods. Other projects that we're doing right now, I'm almost done here. We've got an NSF funded project that is called Spectra. We're looking at the size spectra, that size density relationship. And depending on the slope of that, we can get some information about how productive an area is and how it might be changing out here under different environmental conditions. And then what's the response of all the different plankton to that. This freshwater one, we're looking at, this is rules of resilience. And we're looking again how this emergent populations are changing there and some of the processes. So this is where we're getting into a new environment for us. Working with Francis Chan and a whole bunch of other collaborators on multi-stressor observations, particularly with a focus from us on crab zoea. So for blue crab, excuse me, Dungeness crab, my East Coast. And then we also have a project, a biodiversity observation network, MBOM, with Maria Cavanaugh. We're sort of looking at how to bridge our observations with her satellite observations. And finally, just an idea of where ISIIS has been used. Not all our studies, but with different colleagues who have, there are about 5 or 6 of them, 7 of them out in the world now. Our babies are spreading. They're spreading their wings. So there's a lot of different stuff going on with that. So finally, as I said, acknowledgements. There's a whole lot of people that have been working on this. I've in the development, the physical development, the computing development, and then all the different projects that we've had. This is just a short list of what's going out there. Just so appreciative to have so many wonderful people to be able to work with on different aspects of these students, past students, postdocs, colleagues. It's been a great trip in that regard. So with that, I'll ask if there are any questions.
Thank you so much for sharing with us. I appreciate that so much. We're going to bounce back and forth between folks online and folks in the room for questions. And I can see we've got a question online. Go ahead, Roseanne.
Move forward
Yep.
out of that.
Great
box, right?
job, Bob. Do you have any insights beyond the rest of us about how dearly funded science of academic research programs is likely to roll out under the new administration? A pass will be understood too.
What was that last part?
Oh, a pass will be understood.
Yeah, we all know this is a tremendously challenging time for us. 10 years ago, I would have said funding is extremely challenging just to get, and yet I've done really well with it, better than I even have a right to, I think. But what our current science situation right now is really frightening. And all I can hope is that as momentum is building, that we're going to start getting some traction. I'm hoping I'm seeing the start of a wave of resistance to the changes that aren't being thought well through and come to a better place. So
Thank you, Bob. Questions in the room? All right, John.
Early on you showed beautiful satellite images of ocean temperature spectra in the Atlantic. And of course, for satellite data, there are more things. For instance, they're now measuring vertical migration of zooplankton from satellites. Are you working? I don't know, are you coordinating? Is that useful to you? Are you able to use those kinds of things? Obviously, you're developing a technology.
So
questions. Yeah.
Great question. The M-BOM project with Maria Kavanaugh. Maria has been working with satellite imagery, predominantly looking at color imagery for which would be primary productivity, though they can separate out to different sources. And she's developing a algorithm that defines, I think, about 15 different seascapes, kind of like landscapes and biomes that we have on land. So what we're doing there is trying to first validate those. She's validating it with a flow cytometer to measure just the plankton itself that's being, pigments of which are contributing to that satellite imagery. But with ISIIS, we're sampling the water column underneath it to see how far down it continues to correlate with that. And so what is that seascape telling us about what's going on down deeper? So that at that point, that's where we're not doing a lot of stuff with the vertical migration at this point. That's kind of a neat aside from what we're doing. But it does have relevance when we're thinking about larval transport, because as larvae move between different layers, they can be going in really different directions. So they might be up here, taken offshore when they're small, they come down here, and they get carried back onshore. So that would be it. But I think the diel vertical migration they're looking at is things like euphausids. So.
All right, we've got another question in the room, but I'm going to try to wiggle between rows here, everybody. And I know we're getting close on time, so we'll take one more question here and then we'll wrap up.
Thanks, Bob. I'm curious if, now that you have this, if you've abandoned the net and if you ever still want to have the larvae in hand, and if so, how, what you've seen through the ISIIS and the the scale issues, has it changed your philosophy? So I assume you do use nets still from time to time, but change your philosophy about how you apply the sampling of the net to make it make sense with the blind scale resolution stuff you're getting from ISIIS?
Good question. You have to have samples in hand. If I said no to that, Sue would kill me. But you know, we need it to verify IDs. We need it to have genetic information, age and growth, diet. You know, there's a lot that's really critical. So yes, we would continue that. But your second part of your question is, would we change how we use the nets? Absolutely, we change it, because now we don't need to sample quite as densely in order to describe a distribution. We can describe the distribution with ISIIS, and then we can more targeted use the net to get the samples, specific samples that we need. And so that is one thing that's changed, and it's made our life a lot easier in the lab.
Great. All right, everybody. I think we're going to wrap up there. How about one more round of applause for Bob? All right, everyone. Thank you so much for being here. Hopefully we see you next month for our amazing talk about sneaker waves, which will be fun. So come on back. Thanks everyone for being here. Thanks everyone online.